Welcome students to what is probably one of my favorite chapters in A&P1, probably my favorite body system altogether actually, and that's the nervous system. This is chapter 12, which is going to focus on the central nervous system, which consists of the brain and the spinal cord. We'll spend most of our time talking about the different structures of the brain, and then we'll talk about some kind of high-level cognition, and then we'll talk here at the end about the end of the chapter about the spinal cord. The brain is really amazing. It does literally thousands of things a day, even every minute of every day. So it's really just a phenomenally complex organ that is just incredible in the sheer amount of stuff and information that it can process. So if we look at the brain and the spinal cord, then we are really looking at our central nervous system. So the spinal cord is down here. It travels, of course, through the vertebrae down our back. Where it connects to the brain is an area of the brainstem, shown here in green. The brainstem is sometimes known as the lizard brain because a lot of our autonomic kind of functions get processed here in this kind of brainstem area. It's really vital for essentially keeping us alive. The middle region of the brain here is an area known as the diencephalon, plays several really important roles, also works as a big relay station for us. The back of the brain is the cerebellum, which is a motor kind of balance center, and then the biggest mass of our brain is the cerebrum, or the cerebral hemispheres. And we'll talk about kind of each of these different areas, the cerebrum, the diencephalon, the cerebellum, the brainstem, and then the spinal cord, as we move through this particular chapter here. But before we get to kind of the parts of the brain in a little bit more detail, we have to talk about what really is how the central nervous system is organized in terms of where our neurons are and where their processes are. So white matter and gray matter refers to where do we find the cell bodies of the neurons and where do we find these myelinated axons. So the gray matter is the cell bodies of all of our neurons, and then any kind of really short, maybe non-myelinated kind of axons and neurons. The white matter is then all of the myelinated axons. And so, for instance, if we look at the spinal cord, the gray matter is this kind of inner H or butterfly, and then the white matter is this outer portion here. So when we're looking at a cross-section of the spinal cord like this, we're looking at cross-sections through the myelinated axons out here in the white matter, and cross-sections through the cell bodies of our neurons in the gray matter. And so when we're looking for kind of cell bodies, we're only going to see them really in the gray matter. So the spinal cord, gray matter interior, white matter exterior. The cerebrum and the cerebellum have an outer gray matter that we call the cortex, and then some pockets of inner gray matter as well that we call nuclei, clusters of cell bodies in the central nervous system. And then the white matter is more interior, connecting the gray matter from here, for instance, to gray matter here, or perhaps even to lower. If we look at the brainstem is lots and lots of kind of interior located nuclei kind of embedded in the white matter tracks. And so we see this in all of the regions of the central nervous system, this organization of our neurons into white matter and gray matter. Gray matter is the cell bodies, white matter, mostly myelinated axons. Because the brain is so incredibly vital, it has several important features that keep it protected and safe from harm. The first of which, of course, is just the fact that it's encased in our cranial kind of vault. It's got that hard skull around it, which makes a great kind of hard covering to protect the soft brain tissue. It also has a series of membranes that we call the meninges. If you've ever heard of meningitis, that is inflammation of these kind of membranes around the brain. Then there's something called cerebrospinal fluid, which is made by one of our microglial cells, the ependymal cells. They actually make this kind of blood plasma-like fluid that basically acts like a big cushion for the brain. And then specific to our brain here is a very special barrier that surrounds all of the blood vessels that are in the brain that protects the brain tissue. So if we look at the skull and the meninges first, you can see here is, here's our skin, but here is one of the bones of the skull, and then we've got brain tissue, of course, down here. So the meninges themselves are a series of membranes. There are 3 of them, and so you'll have a membrane and then like a submembrane space and then another membrane and then another submembrane space and then finally the brain tissue. So the outermost membrane is the dura mater. It is a double-walled membrane. It's really leathery. It's very hard. Not hard as in like bone, but it's pretty tough. So it's very protective and encases the whole of the brain. Just underneath the dura mater is then this subdural space, which gets filled with CSF. Then we have the middle meningeal layer, which is the arachnoid mater, which, if you guessed by just looking at the name, arachnoid spider, it might look somewhat web-like, which it does. So it's not quite as tough and leathery as the dura mater. It is much more kind of fibrous and web-like. Underneath the arachnoid mater is then the subarachnoid space, and then we get to the last of the meninges, which is the pia mater, which is really more kind of fine areolar type tissue, very richly vascularized. You can see all the blood vessels kind of bound up in the pia mater. And so that's really what we're looking at when we're looking at these meninges. And if you have ever known anybody who has ever had meningitis, my niece did, it was an inflammation of these meningeal spaces, which typically then puts pressure on the brain and can sometimes result in trauma to the brain. We also have these spaces in the brain that we call ventricles. And the ventricles themselves are also filled with CSF. Each of the ventricles contains a structure called the choroid plexus, which is what actually contains our ependymal cells that make the CSF. And we have 4 ventricles. The first 2 ventricles are the lateral ventricles. They are then separated by a membrane that we call the septum pellucidum. The lateral ventricles are connected to the 3rd ventricle via this interventricular foramen. The 3rd ventricle is encased by the diencephalon. And then the third ventricle connects through the cerebral aqueduct here to then the fourth ventricle, which is here between the cerebellum and the brainstem. So essentially, we have taken neural tissue out of the brain, and we have filled it with these ventricular spaces that then get filled with CSF, which somewhat seems counterintuitive, right? Like you'd think we want to pack as much in there as possible in terms of neural tissue, but the CSF actually has one great feature in that it makes actually has several great features, but one of the big advantages to having these ventricular spaces filled with this fluid is it actually makes your CNS much more buoyant. Imagine if your head felt literally nearly 100 times heavier than double what it currently feels. So the CSF gives a lot of buoyancy to our brain, so it makes it a little bit lighter, not quite so much weight there in the skull. It also basically acts like a big old cushion, big old watery cushion balloon around the brain and the spinal cord as well. It also nourishes the brain. There's lots of nutrients in it. In fact, it has a very similar composition to our blood plasma. A little less protein, different ion concentrations, but still pretty similar to blood plasma. And because of that, it can also carry chemical signals from kind of one part of the CNS to another. We do maintain a constant volume of it. The structure in the CSF, in the central nervous system that makes the CSF is called a choroid plexus. They are found in all of the ventricles, and in those choroid plexuses, we find ependymal cells. So let's take a look. So here's a choroid plexus. You can see it's filled with ependymal cells. They've got their nice little cilia. So the ependymal cells actually make the CSF, and then the cilia do their little beat back and forth motion, and that's what actually circulates and moves the CSF through not only the ventricular spaces, but all of the submeningeal spaces as well. So we're looking here at choroid plexus is here and here in the ventricles. That's where the CSF is made. So not only does it then travel through all of the ventricular spaces, but it's then going to travel through the subarachnoid and subdural spaces all around the brain. And then because the central canal of the spinal cord is continuous with the fourth ventricle here, CSF actually then continues down and circulates not only around the brain, but around the spinal cord and down the middle of the spinal cord as well. So that is our very important CSF. The last kind of bit of kind of brain protection is this blood-brain barrier. Part of the reason the blood-brain barrier is so important is that we really need a very, very stable environment for our brain to function properly. We don't want anything that gets into your blood can immediately go systemic wide, because practically every part of your body is touched by a blood vessel. But we need to be really selective about kind of what gets out of the blood and into the brain tissue. We want to protect that brain tissue as much as possible. There are metabolic wastes that are like a normal waste product for our body, but we don't want those waste products, which are usually transported out of the body via blood, via filtration and urination, but we don't want that stuff getting in our brain tissue. So we have this very specific structure that we refer to as the blood-brain barrier. Unfortunately, this makes a problem when we're trying to treat neurological conditions, because we have to design and manufacture medications that can pass the blood-brain barrier. one of the ways that we can do so is to make them fat soluble, lipid soluble. That can just escape the blood-brain barrier. That's why alcohol affects you very quickly. It's why most anesthesias can affect you pretty quickly because we usually make them fat soluble. So the blood-brain barrier encompasses all of the blood vessels in the brain. With some exceptions. There are a couple of areas in the brainstem and in the diencephalon, which is the hypothalamus, that like actually monitor the chemical composition of the blood, and so they need access to the blood more easily, and so we don't have the blood-brain barrier in those areas. So this is what the blood-brain barrier looks like. You can see, so here we are in a blood vessel. We're in a nice small little capillary, and you can see essentially surrounding the capillary are what we call these foot processes from our astrocytes, right? One of our neuroglial cells. The blood vessels themselves, the walls of the blood vessels, have incredibly tight junctions. So it's hard for stuff to escape anyway, but then we usually have this really thick kind of basement membrane, basal lamina of connective tissue, and then we have these foot processes from the astrocytes. So for something to get out of the blood and into the brain tissue, it has to escape through these tight junctions and through it has to be able to pass these foot processes. And then the foot processes themselves, not only do they make a nice little barrier, but they actually secrete a chemical that makes the tight junctions even tighter. So that is what the blood-brain barrier looks like. Definitely an incredibly important thing that helps keep your brain safe. We're going to stop right here for right now. We're going to pick up with talking about some kind of functional anatomy of our brain structures.